Welcome to today's lesson on the uses of monoclonal antibodies. In this session, we will explore the various ways in which these incredible molecules can be utilised and discuss their advantages and disadvantages. Our lesson for today includes describing some of the specific applications of monoclonal antibodies, such as in diagnostics, the treatment of diseases, and in medical research. We will also evaluate the benefits that these antibodies offer, along with the potential drawbacks that are associated with their use. So far, you have learned about how monoclonal antibodies are produced, in a process that involves the fusion of mouse lymphocytes with a type of tumour cell, in order to create hybridomas that can produce these antibodies continuously. This production method has provided a foundation for the use of monoclonal antibodies in various scientific and medical applications. Building on your previous understanding of how monoclonal antibodies are produced, we will now delve into their practical uses and implications. This knowledge is vital as it enhances our overall understanding of how we can harness biological processes for diagnostic and therapeutic purposes, potentially leading to significant advances in modern medicine. Let's get started. Monoclonal antibodies are lab-created molecules that are engineered to serve as substitute antibodies that can restore, enhance, or mimic the immune system's attack on cells. Monoclonal antibodies are designed to bind to specific proteins on cells or chemicals, making them highly specialized tools in medicine and biochemistry. Let's explore four major uses of monoclonal antibodies, as well as their advantages and disadvantages. Firstly, monoclonal antibodies are instrumental in pregnancy tests, specifically in targeting and binding to a hormone called HCG, human chorionic gonadotropin. HCG is a hormone found in the urine of pregnant people. The part of the pregnancy test stick that people urinate on contains mobile antibodies. These antibodies can move, are complementary to the HCG hormone, and have blue beads attached to them. The test strip section of the pregnancy stick also has immobile antibodies. These immobile antibodies cannot move, but are also specific to the HCG hormone. If the person is pregnant, when they urinate on the stick, the HCG that is present in their urine will bind to the mobile antibodies that are attached to the blue beads. The urine will then carry both the HCG hormone and the blue beads along the stick, where they will bind to the immobile antibodies that reside on the strip. They bind because these immobile antibodies are also complementary to the HCG hormone. This binding process sticks the blue beads onto the test strip, which turns it blue. This indicates that the person is pregnant. However, if the person is not pregnant, then the mobile blue beads will move up the stick, but they will not be able to bind to the immobile antibodies on the test strip, since the person's urine would not contain any of the complementary HCG hormone. In this case, the blue beads would not bind to the test strip, and therefore it would not change colour. This indicates that the person is not pregnant. Monoclonal antibodies can also be used to treat various diseases. One example of this in particular is cancer. This is because monoclonal antibodies can be engineered to target tumour markers, which are antigens found on cancer cells. To begin, monoclonal antibodies are created to bind to specific tumour markers. Drugs that are usually used to treat cancerous growths, such as radioactive materials, toxins, or substances that inhibit cancerous cell growth, can then be attached to these targeted antibodies. The monoclonal antibodies can then home in on cancer cells and bind to the tumour markers, delivering the attached drug directly to the cancer cells without affecting any of the normal cells that aren't targeted by the monoclonal antibody. This targeted approach kills cancer cells whilst minimizing damage to healthy cells. Monoclonal antibodies can also measure and monitor hormone levels or be used to detect pathogens in the blood. Specifically, monoclonal antibodies bind to certain hormones or chemicals in the blood. By binding to these substances, monoclonal antibodies can be used to accurately measure their levels. Monoclonal antibodies can also be used to identify particular pathogens in the blood. This technique is often used to monitor various ongoing conditions and can be used to ensure that patients receive the appropriate treatment. Monoclonal antibodies can be used to detect specific molecules within cells or tissues by binding them to a fluorescent dye. In this case, monoclonal antibodies are tagged with a fluorescent marker. When the monoclonal antibodies bind to their targets, the fluorescent markers help to easily detect them under a microscope. This process is valuable in research and diagnostic labs for identifying the presence of specific proteins or other molecules within cells or tissues. Let's now explore the advantages of using monoclonal antibodies. One advantage is in their specificity. Monoclonal antibodies are highly specific to their target, thus reducing the likelihood of any off-target effects. A second advantage is found in their effectiveness. Monoclonal antibodies can deliver treatments precisely to disease cells. This makes them especially effective for conditions such as cancer. One final example of the advantages of monoclonal antibodies is their reduced side effects. Compared to traditional treatments, such as chemotherapy, monoclonal antibodies generally have fewer side effects. This is because they are able to target specific cells. However, this technology still has some challenges at present. One challenge is found in the onset of certain side effects. Some side effects, such as fever, vomiting, and low blood pressure, are more common than initially expected. A second is their production complexity. Creating specific monoclonal antibodies is challenging and also expensive. Finally, one drawback to monoclonal antibodies at the moment is their limited scope for use. Due to their side effects and production difficulties, monoclonal antibodies are not yet as widely used as we initially hoped. In today's lesson, we explored the uses of monoclonal antibodies, as well as their pros and cons. Monoclonal antibodies are lab-engineered molecules that target specific antigens, thus helping the immune system to neutralize threats. Monoclonal antibodies are produced from identical immune cells. This ensures that they have uniformity and specificity. We discussed the applications of monoclonal antibodies in detecting pregnancy, cancer therapy, and detecting specific molecules in cells, tissues, and the blood. This highlights the ability of monoclonal antibodies to target tumours, deliver drugs, and recruit immune cells. Advantages of monoclonal antibodies include their precision, which reduces harm to healthy cells, and their consistent production. However, they can be expensive, time-consuming to produce, and may cause side effects, with effectiveness varying between individuals. Understanding these aspects helps us to appreciate the role of monoclonal antibodies in modern medicine and the challenges that they present in treatment development.